So now we're going to do protein synthesis in prokaryotes, since obviously their process is a little bit simpler because they do not have membrane-bound organelles or a nucleus. So for prokaryote versus eukaryote genes, prokaryotic cells have their DNA just floating in the cytoplasm. It's a circular chromosome and a very naked DNA, okay? It does not have introns. Whereas eukaryotes, they have their DNA in the nucleus. Their chromosomes are linear, and it's wound up on histone proteins, and they have introns and exons. So for prokaryotes, transcription and translation are simultaneous, okay? So they happen at the same time. So you can kind of see that the DNA is in the cytoplasm. There's no mRNA editing, okay? So the ribosomes read the mRNA as it's being transcribed. So as it's being transcribed, they basically translate at the same time. So for the differences between eukaryotic and prokaryotic, time and physical separation between the processes. For eukaryotes, it takes about 1 hour to go from a DNA to a protein with but for prokaryotes, there's no RNA processing. So there's no double checking to make sure that it's okay with, the splicing and everything like that. So these are some of the mutations that can happen. We have different types. You have point mutations. That's just one individual base. You can have base pair substitution. So you can have a silent mutation. So yes, the bases change, but there's no amino acid change. That's called a silent mutation because it doesn't actually end up changing your amino acid sequence. You can have missense, where it actually does change the amino acid sequence, and a nonsense mutation, where it changes the amino acid into a stop codon. So a point mutation one change of a base can turn into sickle cell anemia. So something as simple as that can become a lethal disease. You can have a frame shift mutation, which we briefly talked about. So you're shifting the reading frame. So it changes everything downstream. You can have insertions, so you're adding bases. You can have deletions where you're removing bases. And then you can have substitutions also. But deletion in diseases, or deletion in your sequences, can lead to a disease called cystic fibrosis. Okay, so if you delete this section right here on chromosome 7, okay, it's called the CFTR gene. If that's deleted, it can lead to cystic fibrosis. So it's the loss of one amino acid can lead to a disease that kills you early in life. Okay, so that's everything on transcription and translation. Please, 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 if you need some extra help, use the links that I put within the lecture to help watch the animations in the videos, and then contact me if you have any issues. And then complete your online College Board FRQs for practice, okay? We're almost done, guys. You got this. I miss y'all. Bye.